let's introduce the next speaker of the webinar, Andrew Smith from Cleveland Clinic. His talk is on TRPC6 deficiency promotes smooth muscle cell dedifferentiation and proliferation after arterial injury. Andrew, please go ahead.
Thank you very much for the opportunity to present our manuscript on the effects of TRP6 channel deficiency on smooth muscle cell dedifferentiation and proliferation after arterial injury. We have no financial disclosures. Calcium permeable ion channels regulate smooth muscle cell phenotype and contractile gene expression programs in a process known as excitation transcription coupling. Canonical transient receptor potential channel subtype 6, or TRP6, is a non-voltage gated cation channel which is highly expressed in endothelial cells and smooth muscle cells, and as Dr. Martin mentioned previously, has been shown to mediate the effects of angiotensin II on smooth muscle cell contraction. We have previously demonstrated that TRP6 mediates inhibition of endothelial cell migration by lipid oxidation products, as shown on the right. Here, wild type and TRP6 knockout mice are fed standard chow diets or high cholesterol diets and perfusion fixed with Evans blue dye 120 hours after a carotid electrocardiography injury. Images of the explanted carotids are shown here, where the amount of blue staining reflects subendothelial collagen, thus de-endothelialized areas of the vessel. And what one can see in the wild type mice is that high cholesterol leads to an increase in blue staining, and therefore it's associated with decreased endothelial migration onto the injured segment of the vessel. And the effect appears to be blocked in TRP6 knockout mice. The effects of TRP6 and TRP6 deletion on endothelial migration in terms of its downstream effects on intimal hyperplasia and remodeling after vascular intervention is not currently understood. This study was initially undertaken to determine if enhanced endothelial migration in TRP6 knockout mice translated into decreased neointimal hyperplasia following carotid wire injury. We found several unexpected findings initially in this study that weren't consistent with our hypothesis. Here we demonstrate that there were histologic and structural differences in the common carotid arteries of wild type and TRP6 mice at rest even prior to injury. But one can see these are 8 to 10 week old male mice, common carotid cross sections that have been H&E stained. And what one can see is that there's an increase in the luminal diameter of TRP6 knockout common carotid arteries and a decrease in medial thickness of TRP6 knockout common carotid arteries. It's difficult to make out on the H&E stain, but in the manuscript, we confirm this more clearly with a resource infusion stain that TRP6 knockout media also contain a reduced number of elastin lamina compared to wild type common carotid arteries. To assess the effects of TRP6 on arterial healing after vascular intervention, we implemented the carotid wire injury of arterial injury. Here, wild type and TRP6 knockout mice were subjected to carotid wire injury and euthanized at 28 days. The injured common carotids and uninjured common carotids were then evaluated by H&E staining. What one can see histopathologically is that TRP6 knockout mice demonstrate much more severe injury phenotypes compared to wild type mice, with luminal stenosis and the accumulation of loose granular tissue or true neointimal hyperplasia. In cohorts of 9 wild type mice and 9 TRP6 knockout mice, one can see that the luminal caliber was essentially preserved after injury in wild type mice, whereas it significantly decreased in TRP6 knockout mice. We also demonstrated that in TRP6 knockout mice, there was negative remodeling of the external elastic lamina, which was not found in wild type mice. We further assessed if cell proliferation was different between TRP6 knockout, injured TRP6 knockout common carotid arteries and wild type carotid arteries. Here, H&E stained common carotid arteries of wild type and TRP6 knockout mice after injury were subjected to cell counting. We just counted the number of medial and intimal nuclei. And what we found was that there was no significant difference in the number of cells in wild type common carotid arteries, but that following injury in TRP6 knockout common carotid arteries, there was a significant increase in cell number. We further assessed if active cell proliferation was present 28 days following wire injury using immunohistochemistry against Ki67. And what we found was that only in injured TRP6 knockout mice were there Ki67 positive nuclei, suggesting that in TRP6 knockout mice, there was evidence of ongoing cell proliferation after injury. We then assessed if there was a difference in the phenotypic state of medial smooth muscle cells between wild type and TRP6 knockout mice, as our results suggested that perhaps there was an increase in phenotypic modulation and dedifferentiation of smooth muscle cells in TRP6 knockout mice compared to wild type mice. To assess this, we took common carotid arteries 28 days after injury and performed immunohistochemistry against smooth muscle actin. What one can see in these representative images is that there's essentially similar staining of smooth muscle actin in the media between uninjured and injured wild type common carotid arteries. But that after injury in TRP6 knockout common carotid arteries, there's a significant decrease in the density of staining of smooth muscle actin. And we evaluated this semi-quantitatively in cohorts of 3 wild type and 3 TRP6 knockout mice, which agreed with this observation that there was a decrease in the intensity of smooth muscle actin staining in medial smooth muscle cells following injury. These results suggested that TRP6 may have effects on arterial remodeling and the development of neointimal hyperplasia, aside from its effects on endothelial cell migration, and that TRP6 may actually have an effect on intrinsic smooth muscle cell phenotype. We therefore modified our hypothesis and posed that TRP6 is required for maintenance or re-expression of a contractile smooth muscle cell phenotype to reduce phenotypic switching and luminal restenosis following arterial intervention. That these are global and congenital knockout mice sort of impaired our ability to test this using just an in vivo model. So to determine the effects of TRP6 depletion on smooth muscle cell phenotype, we isolated, isolated primary aortic smooth muscle cells from thoracic aorta of wild type and TRP6 knockout mice. So these are just in culture, looking at these are proliferation assays of wild type and TRP6 knockout primary aortic smooth muscle cell. In panel A, cultured smooth muscle cells, these are passage 6 to 8, were plated and cultured in 10% FBS with DMEN over a 96-hour time course. And we found significant differences in the growth curve between these populations, with a significant increase in the number of cells in TRP6 knockout primary smooth muscle cells in 96 hours. B is a slightly different proliferation assay, where wild type and TRP6 knockout primary smooth muscle cells are serum starved overnight and subjected to stimulation with PDGF or 10% FBS. We found a slight, though not statistically significant, increase in the response of TRP6 knockout cells to PDGF. This is just counting cell number compared to wild type cells, and a significant increase in cell number of TRP6 knockout primary smooth muscle cells compared to wild type smooth muscle cells stimulated with 10% FBS. In addition, we assessed if migration was different between wild type smooth muscle cells and TRP6 knockout smooth muscle cells. These are razor scrape assays of wild type and TRP6 of cultured wild type and TRP6 knockout primary smooth muscle cells, and the degree percent wound healing, and thus the degree of migration, was assessed 12 hours after razor scrape assay. And what we found was that there was a significant increase in percent wound closure and cell migration in growth factor stimulated TRP6 knockout primary aortic smooth muscle cells compared to wild type smooth muscle cells. So these results in tandem suggested that proliferation and migration, or neointimal hyperplasia associated type behaviors, seem to be increased in TRP6 knockout primary aortic smooth muscle cells. We further assessed if TRP6 knockout smooth muscle cells were phenotypically different in terms of their contractile biomarkers, and if TRP6 knockout was associated with depletion of contractile biomarkers. These are Western blots of whole cell lysates of wild type and TRP6 knockout primary smooth muscle cells, passage 6 to 8. We find that there's a significant attenuation of protein expression of MYH11 and SM22 in the TRP6 knockout population compared to wild type, and we also showed this at the level of mRNA. TRP6 knockout primary aortic smooth muscle cells are associated with compensatory upregulation of closely related TRP channels, including TRPC3 and TRPC7. So thus, assessing only primary aortic smooth muscle cells, it's difficult to determine the effects of TRP6 depletion alone. The results are confounded by the potential effects of upregulation of these bystander pathways and alternative TRP channels. Thus, to determine the direct effects of TRPC6 on expression of contractile biomarkers in vascular smooth muscle cell phenotype, we performed SI-mediated knockdown of TRP6 in an immortalized murine arterial smooth muscle line, MOVAS. Here, this is 48 hours after transfection with TRP6 SIs or control SIs, and we demonstrate that in the presence of TRP6 siRNA, there was a significant decrease in protein expression of MYH11 and SM22. These results suggest that knockdown of TRP6 is sufficient to induce phenotypic modulation, and that TRP6 has a direct role in modulation of contractile phenotype and contractile biomarker expressions in arterial vascular smooth muscle cells. In summary and conclusion, we demonstrate that TRP6 knockout common carotid arteries show altered resting structure and more severe injury phenotypes in wild-type common carotid arteries, and that isolated TRP6 knockout smooth muscle cells show an increase in intimal hyperplasia-related behaviors, as well as a decrease in the expression of contractile biomarkers. Moreover, acute SI-mediated knockdown of TRP6 is sufficient to induce smooth muscle cell phenotypic modulation, and these results suggest that TRP6 may be an important therapeutic target for stabilization of the contractile phenotype after arterial injury to reduce neointimal hyperplasia. These are references. Thank you. I'm happy to take any questions.
Thank you, Dr. Smith. That's a very comprehensive talk. I just have one short question. Then there are more questions in the chat that I want you to answer when the next talk is on. So do you have any, very briefly then, do you have any idea of what the coupling between the channel, the calcium flux through the channel, and the effects that you see on gene expression in these cells?
Thank you. We're currently looking at the relationship between TRP6 and TRP6 depletion and the expression of master transcription factors in smooth muscle cell phenotype. We've demonstrated that TRP6 depletion is also associated with the expression of myocardin, and we're currently looking at mechanisms whereby TRP6 may modulate myocardin transcription and myocardin mRNA expression. We've shown that TRP6 regulates both the mRNA level and the protein level of myocardin. In the moment, at present, we hypothesize that TRP6 may regulate nuclear localization of the upstream transcription factor MEF2C, but obviously there are many other interesting calcium responsive candidate pathways that could contribute to these effects and could contribute to the effect of TRP6 on expression and activity of myocardin.
Thank you very much. We need to move on, but please look in the chat. There are more questions for you.
Yeah, absolutely. Thank you.